Muhammad Umar Farooq Biological Video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork perspective. So in this video we will talk about the basically electron transport chain and chemiosmosis in chloroplast, light reaction and photosystem 1 and 2, which that is the basically the electron transport chain which that are produced by the light falling on the thylakoid membrane. So now we will discuss about in the detail. So here is the cross section of the leaf, which that is the cross section of the leaf is here. That is the meaning of the cross section of the leaf. Is the epidermis is here, and here is the palisade mesophyll cells and spongy mesophyll cells are present is here. There's a palisade mesophyll cell and spongy mesophyll cells is very special character. They contain chloroplast. Chloroplast is a organelle. For example, here I draw a palisade mesophyll cells which that contain cytoplasm, nucleus, vacuole, cell wall, and chloroplast. There's chloroplast is basically 100 to 200 chloroplast in the single cells are present, which that are bigger organelle. It means double membrane organelle, and it means the quantity of the chloroplast is less as compared to the another organelle which that are shortened and smaller. So here is the chloroplast. Now we will discuss about the chloroplast. That's chloroplast contain thylakoid membrane, which that is the granum. It is known as grana, plural grana, and is the outer membrane is the inner membrane, and inside the outer and inner membrane is thylakoid are present, and stroma within the thylakoid which that are floating the thylakoid into the stroma, and the stroma is the jelly-like fluid structure fluid which that are present between the thylakoid, and thylakoid is made up of here is the stack-like structure that's contain a lumen. That's inside is lumen, and there's basically grana. That's grana is single. We will discuss about the granum, and is the lumen. The lumen inside is the thylakoid membrane, which that lumen contain different types of enzyme and proton gradients. It means that too much proton are present into the lumen due to the hydrolysis or NADPH, nicotinamide adenosine nicotinamide adenosine dihydrogen is the basically breakdown of the structure due to that. So here is the chlorophyll, which different types of pigments, yellow, red, orange. Carotenoids are present, carotene orange basically, and xanthophylls yellow to orange is color. So here is the chlorophyll structure, which that are magnesium is the central unit, and for the chlorophyll A is known as methyl group addition in the green color. You should know about that. And the B color is the hydrocarbon color, basically oxygen and carbon and hydrogen. Is the tail and head. He is the head. Basically head is used for the absorption of the light. That is the magnesium is the central part of this molecule, and here is the basically phytol hydrocarbon tail are present. That's so here. So here is the photosystem which that are contain chlorophyll molecules. That chlorophyll molecule is known as the composition of chlorophyll molecule in the sheet of the thylakoid membrane is known as photosystem. With that molecule pigment is known as antenna pigment molecules or antenna complex it is known as. And here is the primary acceptor, with that accept the electron after the jumping of the electron molecule to molecule, and the 680 picometer is the basically light absorption, and that's electron will be excited. So here I will discuss about the chloroplast which that are present is here, and there's chloroplast is basically when light will be fall into the chloroplast. There's thylakoid membrane is here and absorb the thylakoid membrane light, and in the presence of water photosystem 2 absorb first, an electron transport chain will begin, and photosystem 1 will be accept the electron, and there's the light reaction will be give the, in the case of energy, ATP and NADPH. There's ATP and NADPH will be enter into the Calvin cycle, that is known as dark reaction. That's Calvin cycle is dark reaction, or basically that's dark reaction absorb the NADPH and ATP. That's NADPH and ATP will be produced the sugar, that's in the form of sugar is the glyceraldehyde 3 phosphate, which that are further converted into by the different process into the sucrose and starch, basically amino acid and fatty acid, some other stuff. So it's a thylakoid membrane which that contain lumen, and we will discuss about the here is the membrane of the thylakoid. So the thylakoid membrane is contain different type of enzyme and different protein, globular protein which that are present in the form of channels, like for using for the moving the electron from photosystem 1 to photosystem 2, and there's the many enzyme are also present. Now we will discuss about the transporter proteins which that are also present, which that transporter protein is known as, here is the photosystem 1 to go cytochrome complex. Okay, so cytochrome complexes contain different types of elements. So first of all we will talk about the photosystem. So photosystem is composed with the photosystem 2, basically when light will be dispersed and light will be fall on the photosystem 1, basically sorry photosystem 2. Then photosystem 2, that magnesium is the central element which that absorb the light, and there's the absorption of the light in the form of, and there's a light will be absorbed and excite the electron due to the hydrolysis. There's water will be hydrolyzed into the 2 electron and 2 proton as well as oxygen molecule. One molecule of oxygen O2 is responsible for the 2 hydrogen 2 water molecule hydrolysis. So it means it's a half. So is the 2 electron will be produced, and this 2 electron will be go into 4Mn, that's the first acceptor 4Mn, which that you should remember it, which that is a protein part which contain a central units of 4Mn, that is accept the 2 electron. Okay, alright. So there's exception of the 2 electron we go inside the photosystem 2. There's inside the photosystem 2, there's an electron will be accept, and there's another protein molecule that is the TYRZ is accept the electron and give farther way to a special pair of magnesium. There's a special pair of magnesium ions with that accept the electron further, and that exception of the electron is the go into the another proteins that is known as phenophytin proteins, and there's phenophytin protein will transfer the more to the plastocoanin A. That's a plastocoanin A and plastocoanin B. That's transformation. There's a plastocoanin A will be accept the electron, 2 electron, then basically, and there's plastocoanin B will be accept the electron further by protein exception of electron, and it means the basically outer side of the lumen, which that are outer side of the cell membrane, there's a proton gradients which that are too much proton are present. So now we will talk about the plastocoanin B will accept 2 proton also, and 2 electron and 2 proton are accept by a plastocoanin B, and then go with the coanin pool which that are another protein, and that's go coanin pool to plastocoanin C. Thus plastoquinine C will accept 2 more proton. It means 4 proton will be addition. Thus 4 proton is very important. I will give you a concept about the 4 proton. So here is the 2 electron and 4 proton added by the plastoquinine C. There's a go into the plastoquinine C. There's a plastoquinine C will pump out the 4 proton in the same time, and that's go into the heme group. There's a heme group will be there's a 4 proton is going upward and 2 electron will accept by a heme group. There's a heme group will transfer the again toward the plastoquinine, and that's plastoquinine again accept 2 proton, and that's go back again. There's a cycle will be continue, and 2 electron which that are accepted by the plastocyanine and different protein which that are copper containing protein which that is accept basically plastocyanine, which that is plastocyanine is transferred the 2 electron into the magnesium special pair, and there's the plastocyanine will go back again for accept the another electron from the cycle will be continue, and there's the continue for using the continue that plastocyanine will back toward the basically cytochrome complex. Look at there, and there's the basically iron sulfur center are present which that are accept the electron by magnesium to quinine K. There's a quinine K will be is here is that is the ferridoxine hormone protein which that are iron containing protein. Both ferridoxine 2 molecule will accept the 2 electron for each, and that's go into the another photosystem. Basically is here is the especially is here is the photosystem 1 which that are accept the electron, and that's the next go into the FADH FAD mononucleotide, and then ultimately go and ferridoxine will be back again, and okay, and no electron will be accepted by the NADPH reductase enzyme. Okay, right, here is the FAD mononucleotide which that are present is here, and here the enzyme are present is known as NADP plus reductase enzyme which that electron enter and go inside outside the lumen in the into the stroma, and NADP plus will be converted into NADPH by the 2 electron using and 1 proton is using. The 2 electron and 1 proton is responsible for the making one NADPH. Okay, right. Now we will discuss about the chemiosmosis. The chemiosmosis is the another discussion about the here is the basically I will draw a ATP synthase channel which that are proton will enter into the A side. That's the there are many sides, A side and B side and C sides, which the C side is using for the rotation, and A B and AB which that are attached with the here is the subunit of the C side which that contain a too much proton which that accept the proton and release the proton in the A side. So A side to release, and here is the cycle will be continue, and that the rotation will be occur. So I hope you make understand about that simple science about that. Here is the proton will add, and here is the release from the proton. So hole will cover the hole by a next, and that cycle will be continue. And here the basically upper side is known as rotator, and he is the stator which that is downward, and beta subunit and alpha subunit and delta subunit and eta subunit are present. So it means the beta ATP synthase, beta subunit is responsible for the ATP synthase by a 4 proton. That's a 4 proton is used for the 1 ATP using. It means 2 molecules of water will be hydrolyzed and give the 2 proton, 4 proton, sorry, and there's the 4 proton is make a 1 ATP. So sometime scientists say that the basically 4 proton or 6 proton, but we will understand about here, and that oxygen will be released from here as a byproduct by the hydrolysis of 2 molecules of water will be 1 molecule of oxygen will be released from the stomata. So here the proton will be enter from his side and here side, and there's the go again, and the cycle will be continue, and 4 proton is used for the ADP to ATP. So that's very easy to understand about it. So the beta unit is used for the ATP synthase enzyme, which that are used for the ATP synthase. So here the cross section, which that are alpha to eta and beta to eta. That's a subunit is that's the upper part, and here is the basically C side and A side as well as B side. The B side is elongated to where the interconnected with the delta ATPase delta stator side. So here is the rotator side, which that rotate and proton bind to release into the stroma, and there's the from lumen to stroma, and there's the releasing of the protons and attached with the ATP synthase from the beta subunit to produce ATP. So that's a simple. So here is basically what I will give you a concept about that things. That here is the hydrogen ion will release into the stroma from lumen. So there's the ATP used from the here side, the beta subunits is that's here. That's the cross section of the rotator and stator. So basically the rotator and stators are present is here. There's a stator mean not rotate, and rotator mean that's rotate, that includes C side and gamma side and a delta side and eta side, and stator mean A side, B side and delta side A and eta and beta. That's the equal, that's the subunit of the this thing. That's contain the basically C side contain the amino acid because it is made up of protein, and that's the a single unit of amino acid. I will give you a concept about simple understand about the here. So here is the carboxy group, which that is attached with the carbon to carbon bond, but it's okay. But here is the oxygen and here is the oxygen, but here is the hydrogen that's attached with the carbon. That's present in the carboxy group. So it means the covalent bond, okay, right, and that's covalent bond is present is here, and there's the proton mean hydrogen which that are in the form of proton, and they're released into the stroma, okay, and there's the release here is the from carbon another chain, and is the release of the carbon from the carbon to proton. That is proton is go into the stroma, okay, and then again will be add from the lumen side. From the lumen there's a basically from lumen add more and release, add release, add release. That is the cycle will be continue. So I hope you make understand about the that things which that are which that is the basic importance. So here is the basic important. This hydrogen ion will cover the whole in the amino acids in after the pumping proton. It means the pump proton from here to here is downward, here to there's the here is the hole will be produced, and that's covering for the hole is add the proton. That is the protein, the amino acids addition. So thanks for watching. Please subscribe channel and comment about this video.